Hi everyone. In this video, we will be talking about Epstein-Barr Virus serology. Let's first take a look at the different characteristics and viral structures of your Epstein-Barr virus, otherwise known as your EBV. EBV is a member of the herpes viridae family of viruses. This viral family is composed of large, complex DNA viruses surrounded by a protein capsid and an outer envelope. In this figure, you can see the different structures specifically of your EBV. The genetic material of this virus is a double-stranded DNA, and this is surrounded by a protein capsid. Collectively, this structure is known as your nucleocapsid. Now, the nucleocapsid is encompassed by your tegument, which is in turn encompassed by an envelope. Our EBV is an envelope virus, and on this envelope, you can see various envelope proteins. We need to take a look at these different structures because they will come into play later on when we discuss the different antibodies that we have against our EBV. Let's next take a look at how the EBV is transmitted. The most common mode of transmission is contact with salivary secretions. This can happen in a variety of ways, such as kissing and even sharing of eating utensils. Other less common modes of transmission include blood transfusions, bone marrow transplants, solid organ transplants, sexual contact, and perinatal exposure, which here means transmission from the mother to an unborn child. Let's now talk about the epidemiology of our Epstein-Barr virus. When you are infected with this virus, it actually confers to you lifetime immunity. However, small populations of the virus remain viable in the body, thus an infected person is a possible carrier for life. The age group at which you will be most likely infected with the Epstein-Barr virus depends on where you live in the world. In developing countries and in lower socioeconomic groups, the infection usually occurs in early childhood. Meanwhile, in industrialized nations or those areas that have a high sanitation standard, infections usually occur in adolescence or adulthood. Now, our Epstein-Barr virus is one of the most common viruses that infect humans, and it is estimated that approximately 95% of adults have been infected, and this is based on the serodata of these adults. That means that 95% of adults have antibodies against our Epstein-Barr virus. Let's now take a look at the different diseases associated with your EBV. The most common disease is infectious mononucleosis, otherwise known as mono or kissing disease. Other diseases include your lymphoproliferative disorders like Burkitt's and Hodgkin's lymphoma, and even non-hematologic malignancies like your nasopharyngeal carcinoma and gastric carcinoma. It can also cause lymphoproliferative disorders in immunocompromised patients. For example, in patients with AIDS, you will often notice that they have nervous system lymphomas that are associated with your EBV. And they can also cause post-transplant lymphoproliferative disorders. Now in this lecture, we'll be focusing primarily on infectious mononucleosis. This disease has different clinical manifestations depending on the age group of the infected individual. In infants and young children, they are usually asymptomatic or mild, and in healthy adolescents or adults, this is where you see your symptoms. Now the classic symptoms of your IM or infectious mononucleosis is fever, lymphadenopathy, this is tenderness and swelling of your lymph nodes, and sore throat. These symptoms usually last 2 to 4 weeks. However, some other symptoms like fatigue, myalgias, and the need for sleep can persist for months. In this figure, you can see the pathophysiology of your infectious mononucleosis or IM. So infection or acute infection usually starts in the oropharynx, and the first cells to be infected are your epithelial cells. The virus binds to B1 integrin on these epithelial cell surface, and the epithelial cells phagocytose the virus. Once inside the cell, the virus then begins its life cycle. So it penetrates the nucleus of the cell and forces the cell to create more copies of the virus. Once there are enough copies, the epithelial cells are lysed and the virus infects more cells. So the virus moves to the salivary glands and the tonsils. Now our tonsils are an example of a lymphoid organ. And here in the tonsils, the virus moves on to your B cells. They infect B cells using the surface CD21 markers of your B cells. At this stage also, B cells starts to produce a variety of antibodies. And at this stage, the virus is now being controlled by these different antibodies, and the immune system is starting to overcome the disease. However, the virus will still persist in a variety of memory B cells. Now, memory B cells are sort of like immortal B cells that are able to produce antibodies whenever we re-encounter a pathogen that was previously encountered by your immune system. Now, the virus can cause latent infections, and here, the EBV nucleic acid exists as episomal DNA. This means that this DNA of the virus is actually living or existing outside the nucleus. That means that it doesn't affect the B cell function. So sometimes there is periodic activation of this virus, which leads to viral shedding in saliva and genital secretions. So although your partner may not have any symptoms, you will not be able to know if there are already viruses that are being shed in their saliva and genital secretions. And this is one of the main factors why it's very easy to transmit this virus. Now, in the laboratory, patients with infectious mononucleosis will usually present with these different findings. First, we have absolute lymphocytosis of greater than or equal to 50%. This refers to the total number of lymphocytes in circulation. In this population of lymphocytes, at least 20% of them will be atypical lymphocytes, mostly activated cytotoxic T cells. In this figure, you can see some examples of atypical lymphocytes, and these are just lymphocytes that do not have the normal morphology of our lymphocytes. In this first example, you can see that the lymphocyte is very, very large, and its cytoplasm is starting to engulf other red blood cells surrounding it. The nucleus is also very large, and you can see some vacuoles being formed in the cytoplasm. Here we have a sort of normal lymphocyte, but it's atypical because the nucleus is indented and there are some small vacuoles in the cytoplasm. Other laboratory findings include heterophile antibodies and antibodies to EBV antigens. Both of these we'll be discussing in this lecture. Speaking of antibodies, B cells produce these three types of antibodies whenever they are infected with the EBV. First, EBV-specific antibodies. Then we also have heterophile antibodies and our autoantibodies. Among the different autoantibodies, some examples are coagglutinin, rheumatoid factor, and anti-nuclear antibodies. Anti-nuclear antibodies are antibodies that attack nuclei or genetic material within our bodies, and this is the antibody that is elevated in systemic lupus erythematosus. Let's now proceed to our heterophile antibodies. There are three types of heterophile antibodies that we encounter in humans. First are Forceman heterophile antibodies. These are antibodies formed against Forceman antigens, a type of heterogenic antigen discovered in guinea pig tissue and in many other animals. Next, we have serum sickness heterophile antibodies, which are antibodies formed during serum sickness, a type 3 hypersensitivity reaction to animal-derived anti-sera like your horse or bovine anti-sera. Nowadays, serum sickness isn't very common, but can still sometimes be encountered in patients who are inoculated with vaccines containing these animal anti-sera or who are given antibiotic therapy. Lastly, we have infectious mononucleosis heterophile antibodies, which are antibodies that appear in patients infected with the Epstein-Barr virus, and this is the topic in the next slides. So talking into more detail about our infectious mononucleosis heterophile antibodies, they are usually IgM, and they have the following characteristics. They can react to horse red blood cells, sheep red blood cells, and ox red blood cells. Ox red blood cells might also be called your bovine red blood cells. And they can be adsorbed by our beef erythrocytes, but they are not adsorbed by our guinea pig kidney cells. When we say adsorbed, this simply means that these antibodies are taken up by these cells and they are neutralized. Now, our IM heterophile antibodies are found in 40% of IM patients within the first week of infection, and this increases to 80 to 90% of patients by the fourth week. However, these antibodies are short-lived and they usually disappear by 3 months after the onset of symptoms. However, in some individuals, they can be detected up to 1 to 2 years after you've had the infection. Now, heterophile antibody detection is ideal for point-of-care testing, but we must take note that they are not as sensitive or as specific as tests for antibodies towards EBV. And this is because heterophile antibody detection is very susceptible to false negative and false positive results. False negative results occur in approximately 10% of adult patients with infectious mononucleosis, and this increases to 50% if the patient is a child younger than 4 years old. You can also have false positive results in some cases of lymphoma, viral hepatitis, malaria, and certain autoimmune diseases. Let's now proceed to the different methods for detecting our heterophile antibodies. We will first discuss the classical methods or the original methods for detecting these antibodies. First is the paul-bunnell presumptive test. This is a general screening test for heterophile antibodies and is used to determine the titer of heterophile antibodies in patients with infectious mononucleosis. In this test, serial dilutions of patient serum are incubated with sheep red blood cells, and the titer here is the reciprocal of the highest dilution to show red blood cell agglutination. The significant titer is greater than 56. The caveat to your paul-bunnell test is that it cannot differentiate between the different heterophile antibodies, like your Forceman, serum sickness, and your IM antibodies. Therefore, if you have either Forceman or serum sickness antibodies, they will cross-react and cause false positive results. To combat this lapse, the Davidson differential test was developed. This is performed only when the titer for the paul-bunnell test is greater than 56, and the main function of this test is to differentiate between the different heterophile antibodies. The significant titer for this test is greater than 224. The Davidson differential test is composed of three main steps. This is the adsorption step, the addition of sheep red blood cells, and agglutination. In more detail, first we have the adsorption step, which involves the removal of antibodies in the serum. Here, the serum is separated into two portions and treated with different adsorbing reagents, specifically our guinea pig kidney cells and our beef red blood cells. After the adsorption step, the setups are centrifuged and the supernatant is mixed with our red blood cells. Here we are using sheep red blood cells, and we observe for agglutination. Note that guinea pig kidney cells are able to adsorb or remove Forceman and serum sickness antibodies. Meanwhile, beef red blood cells are able to adsorb or remove serum sickness and infectious mononucleosis antibodies. In this table, you can see the adsorption pattern of each of the heterophile antibodies. First, for Forceman antibodies, they are adsorbed by guinea pig kidney cells, but not by beef red blood cells. Meanwhile, serum sickness antibodies are adsorbed by both guinea pig kidney cells and our beef red blood cells. And lastly, infectious mononucleosis antibodies are not adsorbed by guinea pig kidney cells, but are adsorbed with beef red blood cells. So just a recap, once these antibodies are adsorbed by these different cells, they are no longer able to agglutinate our sheep red blood cells. And that brings us to this next table, which shows the agglutination results for each of our different antibodies. So first, for Forceman antibodies, we can observe agglutination in the beef red blood cell treated serum. Meanwhile, for serum sickness, we will not be able to see any agglutination at all in both the guinea pig kidney cell treated serum and the beef red blood cell treated serum. And lastly, for IM antibodies, we will be able to observe agglutination in the guinea pig kidney cell treated serum, but not in the beef red blood cell treated serum. Now, the classical methods that we just discussed are rather tedious. So more recently, this next test called the monospot was developed. This is a rapid slide agglutination method, and its principle is that it tests the ability of serum adsorbed with either guinea pig cells or beef erythrocyte antigens to agglutinate horse red blood cells. So essentially, the monospot is a shortened version of the two tests which we just discussed. Here, instead of sheep red blood cells, the test now uses horse red blood cells, because later on, scientists discovered that horse red blood cells are actually more prone to agglutination than our sheep red blood cells. So this is a slide test, and these are the different results that you will be able to find. So on the left side, you can see the negative results, no agglutination, and on the right side, you can see the different positive results. You can have a weakly positive result, a positive result, or a strong positive result. These are all heme agglutination results. For the procedure of the monospot test, first place 1 drop of patient serum and 1 drop of guinea pig kidney antigen suspension on a glass slide. On another slide, add 1 drop of the patient's serum and 1 drop of beef red blood cell antigen suspension. Then after this, add 10 microliters of 20% horse red blood cell suspension to each slide and mix, and then stand for 1 minute and observe for agglutination. Here are the possible results that we will encounter when doing a monospot test. First, if you see stronger agglutination with sera adsorbed by your guinea pig kidney cells, this is a positive result, and this means that you have heterophile antibodies from infectious mononucleosis. Next, stronger agglutination with the sera adsorbed with your beef cell stroma. This is a negative result, and this means that you have your Forceman antigens, and if there is no agglutination in both mixtures, this is also a negative result and indicates that your heterophile antibodies are from serum sickness. So although the monospot test is relatively easier to perform than the classical methods for heterophile antibody detection, currently it is not being widely used. Instead, we have these alternative methods for detecting our heterophile antibodies. First are the rapid agglutination tests, and the most common one is your latex agglutination. Here, instead of red blood cells, latex particles are coated with the heterophile antigen. We also have immunochromatographic kits, which are also relatively simple to use and very easy to read. Before we can talk about our Epstein-Barr virus antibodies, we first have to talk about our Epstein-Barr virus antigens. Now, the infection of our EBV occurs in 3 phases: the early acute phase, the late phase, and the latent phase. At each phase of the infection, the immune system encounters a variety of antigens. And as they encounter these antigens, they also create or develop the corresponding antibody to these antigens. So first, in the early acute phase, we have EAR, which stands for early antigen restricted, and our EAD, or early antigen diffuse. Now, these 2 antigens get their names depending on where they are found in the infected cell. The restricted antigens are restricted to the nuclei. Meanwhile, the diffuse antigens are found both in the nucleus and in the cytoplasm of the infected cell. In the early acute phase, the virus first enters the cell and infiltrates the nucleus, where it can start to replicate its viral genetic material. And that is why the EAR is the first antigen to be detected by your immune system. After the virus has replicated itself, it then starts to create copies of the different proteins it needs to create the viral particle. And that's the time you will see the early diffuse antigen, again, both in the nucleus and the cytoplasm. Next, we have the late phase antigens. These are the viral capsid antigen, or VCA, and the membrane antigen, or MA. The late phase of infection occurs after viral DNA synthesis, when the virus has already created multiple copies of itself within the infected cell. When it has enough copies, the virus lyses the cell so that it can spread to surrounding cells. So the antigens that we encounter in the late phase correspond to the different parts of the virus, like your capsid and your membrane, or the viral envelope. Lastly, we have the latent phase antigens. In the latent phase, Epstein-Barr virus becomes dormant in our B cells, so the only antigens that our immune system encounters are your eBNA. This is your EBV nuclear antigens, or antigens to the genetic material of your EBV, for which there are 5 subtypes. First, eBNA 1, then 2, 3, 4, and 5. Alternatively, eBNA 3 is also known as 3A, eBNA 4 is known as 3B, and eBNA 5 is known as LP. During reinfections or reactivation of the virus, the immune system then encounters LMP, or latent membrane proteins. There are three subtypes: LMP1, LMP2A, and LMP2B. Here we can see the different techniques used for the detection of EBV antibodies. We have our indirect immunofluorescence assay, or IFA, enzyme-linked immunosorbent assay, or ELISA, chemiluminescent immunoassay, or CLIA, and your flow cytometric microbead immunoassays. We won't discuss them in much detail in today's lecture, but we have prepared another lecture which explains each of these in turn. So you might be wondering, why do we need to detect all these different antibodies towards EBV? One of the main uses for these different antibodies is to tell what stage of infection the patient currently is experiencing, or if the patient has already had a previous infection. In this table, you can see the different stages of infection and a description of what kind of antibodies we can expect in those patients during that stage. First, we can tell that the patient is still susceptible to the disease or has not had a previous EBV infection if they are seronegative. This means they lack any antibodies towards EBV, especially towards our VCA or viral capsid antigen. Next, we can tell if the patient has a primary infection if we detect antibodies towards VCA or the viral capsid antigen. Specifically, the first antibody to be detected is the IgM, which is a primary response antibody. This means that IgMs are the first type of antibody to be produced during an infection. Also, eBNA, or antibodies towards eBNA, are absent, and we will be able to see a high or rising titer of IgG antibodies to VCA and no evidence of antibodies to eBNA after at least 4 weeks of infection. Here, IgG is usually produced in the late stages of infection or after the body has just recovered from an infection. Next, if antibodies to eBNA and increased antibodies to early antigens are present, then we can say that the patient may be experiencing reactivation. Lastly, if your patient has had a past infection of EBV, then you'll be able to observe antibodies to VCA and eBNA. In this table, you can see a summary of all the different antibodies we discussed and their presence at different stages of the disease. Included here are our EBV antibodies and our heterophile antibody. In an uninfected patient, of course, we would expect to see no antibodies at all. These include no EBV antibodies and no heterophile antibodies. During acute infectious mononucleosis, we see an increase in your anti-VCA, specifically your IgM. Again, this is the first antibody that is produced in response to the disease, and IgG. Also, we are able to see our EAD, or early antigen diffuse antibodies, but we are not able to see antibodies against our eBNA. Meanwhile, for heterophile antibodies, we have a variable result. This is because it's very possible that we have a false negative result for our heterophile antibodies. Next, during convalescent infectious mononucleosis, this is the stage in which the patient is starting to recover from the disease. You will still see IgG anti-VCA, but no longer see our IgM. Also, for the anti-EA, we might have a variable result for our EAR, or our early antigen restricted antibodies. And this is the first stage in which you will see our anti-eBNA. And again, for heterophile antibodies, we have a variable result. And this is because, again, it's very possible for our patients to have false negative results for heterophile antibodies. This is why it is preferred that we use our EBV-specific antibodies rather than our heterophile antibodies when detecting the disease. Next, for a past infection of infectious mononucleosis, we can observe IgG anti-VCA and our anti-eBNA. And our heterophile antibodies this time will be negative, because if you can recall in our previous slide, we mentioned that our heterophile antibodies are only present for a few months in most patients. Next, we have our chronic active infection of infectious mononucleosis. This is a rare complication in very few patients in which the EBV virus does not go away or does not reach its latent phase. And here this is characterized by a very high IgG anti-VCA level, variable IgA anti-VCA, and positive anti-early antigens. Both EAD and EAR have antibodies against them. You also have a variable result for anti-EBNA and negative heterophile antibodies. Currently, it is not known why some patients experience chronic active infectious mononucleosis. Lastly, here you can see the different antibody correlation patterns of our different EBV-associated diseases, like your post-transplant lymphoproliferative disease, Burkitt's lymphoma, and nasopharyngeal carcinoma. Alright, so if you wanted to know more about the things we just discussed, you can check out these references. Thank you for listening, and we hope to see you in the next video.